So we've been talking about conductance-based neuron models. We started out by talking about the Hodgkin-Huxley model of the squid giant axon, which is a single compartment model with only a couple ionic currents. And then I explained that it's very easy to add more complexity to this basic model. We can add more currents, and we can include multiple electrical compartments to model the spatial complexity of neurons. So I wanted to give an example from somewhat recent literature about how complicated these models can actually become, and what are some of the issues that we begin to face when we consider these additional complexities. So this is a paper that I'm going to show a couple figures from, and I'll give a link to the PDF. And it presents a computational model of a neuron in the crustacean stomatogastric nervous system. So this is the first figure from that paper, and it gives a schematic illustration of what the model actually is. So there are 4 electrical compartments. There's a compartment that models the axon, the soma, or the cell body, and 2 compartments that model the neurites, or dendrites. So the first thing to notice is that this is a multi-compartment model. There's 4 electrical compartments, so there's 4 membrane potential variables that are being solved for. And in addition to having 4 compartments, there's several voltage-dependent currents. So this is more complicated than the Hodgkin-Huxley model of the squid giant axon, in that there is some spatial complexity, in that there's 4 compartments, but there's also more ionic currents that are in some of these compartments. So for example, there are calcium currents in the soma and neurite compartments. The axon compartment is perhaps more understandable, and it's actually pretty similar to the Hodgkin-Huxley model. There's a sodium current, which has both activation and inactivation gates, a delayed rectifier, potassium current, which is Kd, and that just activates, there's no inactivation. And then there's a final current, which we haven't talked about, which is called the A-type potassium current, which is a potassium current that has both activation and inactivation. So these are just different currents that have been identified in these cells, and they're modeled in the same way as the Hodgkin-Huxley channel models that we have been talking about. So the only difference is that there's just more of them. So this model is, it's definitely more complicated, but at the same time it's somewhat simple. You could imagine having many more electrical compartments as well as more ionic currents. So the idea behind this paper is that this is a model that is relatively simple. It can capture a lot of the complexities of the neurons we're looking at, but it's not overly complicated. There's not hundreds of electrical compartments. There are other papers out there that do look at model neurons with that many, with a very large number of compartments. But this is something that's a little bit easier to understand. So this next figure presents data on the biological neuron that we want to model. So this neuron, as I mentioned, is from the stomatogastric nervous system. It participates in what's known as the pyloric central pattern generator, or pyloric circuit, and it creates a rhythmic muscle movements in the gut of crabs and lobsters that filter food particles. On the left, in panel A, this shows the morphological features of the cells. So remember, we have a 4-compartment model. 2 of the compartments deal with the neurites, which are here, and we also have a somatic compartment, which is the cell body, essentially, and then the axon is down here at the bottom of the screen. So those are the electrical compartments that we have in our model, and you can see that just having 4 compartments does not fully capture all the morphological complexity of this cell. So we are simplifying our model to a relatively extreme extent, but you can still replicate the behavior of the neuron, which is really the focus of this paper. So the behavior of the neuron is shown in panels B and C. The LP neuron, or lateral pyloric neuron, as I said, is part of the pyloric circuit. It receives inhibition from 2 other neurons neuron types called PD neurons and PY neurons. And they both inhibit LP during this period. So this is when PD inhibits LP, and this period here, so this is where PY inhibits LP. And all other times the neuron is active and firing spikes, such as right there. So this is a relatively complicated activity pattern to replicate, and that's why it's interesting to build a model of this behavior. You can see that there's slow oscillations. These slow oscillations are driven by calcium currents, and riding on top of these oscillations are action potentials, which are driven by sodium and potassium currents, in a similar way to the original Hodgkin-Huxley model, although there are some complexities that we're glossing over here. So this here shows the behavioral pattern, or the activity pattern, of the LP neuron in the somatic compartment. Remember that we're modeling the voltage not only in the soma, but in other compartments. We're primarily going to be interested in the pattern of voltage in the soma because that's what's most easy, that's what can be recorded from experimentally. So before I show you the output of the model, I want to take a brief moment to comment on the ion channel models in this paper. And they all follow the same basic formula that Hodgkin-Huxley first pioneered. This table shows the steady state and time constants for the activation and inactivation gates of each channel. And we already know that, for example, for the activation gate, they will change over time according to this equation, which simply means that the variable will approach some steady state, exponentially approach that steady state with some time constant. And the steady state and the time constant are both functions of the voltage. So that's what these equations here. For example, the first row gives the delayed rectifier, the steady state, and the time constant for the activation gate, and it doesn't have an inactivation gate, so those are left blank. But all channels obey this basic formalism, and they have different kinetics and different properties, which have been experimentally measured. But the point is that they all follow this simple equation, and we know how to numerically integrate this equation, and the paper does this using the same method that I've covered in a previous video, which is the exponential Euler method. So if you haven't looked at those previous videos, I would recommend going back just because those details are relatively important. So this figure illustrates an important result that I wanted to emphasize in this video, and that is that even in a relatively simple model, you have so many model parameters that if you search across those parameters, you can find many biologically feasible models of neural activity. So in panel B, the authors are showing the output of 6 example models. All of these models reproduce the activity pattern that is characteristic of the LP neuron in the pyloric circuit. So these are quote-unquote good models. They reproduce what we think the biological neuron is actually doing. However, each of these models has substantially different parameters. And what do I mean by parameters? Well, panel C shows the parameters that were searched over in this paper. Most of them are maximal conductance parameters, such as GKD here. This roughly correlates to the number of delayed rectifier potassium channels in the soma of the neuron. So if you randomize the number of channels, you can find that there's many different combinations of channel parameters that produce realistic behavior. And the basic take-home message is that there are so many parameters, and even though we have a relatively complex pattern that we're trying to reproduce, you can still find many solutions or many different models that, to a first approximation, reproduce the biology relatively well. And this is one of the fundamental challenges of building these complicated neuron models, is that there's so many parameters, and it's relatively difficult to fully characterize a cell's phenotype, that it's difficult to distinguish between which of these parameter sets are reasonable models and which of them are models that are superficially similar but actually completely wrong in a certain sense. So the authors of this paper go on to analyze these successful neuron models in much more detail. So rather than studying just a single example, they study all models that meet criterion sufficient to reproduce the biological behavior. And it's a very nice paper, and I encourage you to read the details of it, because I can't cover all of them in this short video. But as it is, this is sufficient to emphasize the point I was trying to make, which is that it's very easy to develop complicated conductance-based models, and easy to include more ionic currents and electrical compartments. However, these complicated models come with many quote-unquote free parameters, such as maximal conductance parameters for each current. Again, the maximal conductance being loosely correlated with the number of channels in the cell's membrane. Typically, there are many different parameter combinations that reproduce biological behavior, and that's what's shown down here. So it's erroneous if you just picked a single parameter set, perhaps by fiddling with parameters and hand-tuning them, and studying just a single parameter set, and it's probably better to recognize that there's many different parameter sets in your model that reproduce biological behavior. In fact, direct experimental measurements of cell parameters show substantial biological diversity, and this is what I'm showing on the right. This figure is from a different paper, which is trying to measure a lot of the parameters that were varied in this modeling paper down here. So for example, the maximal conductance of the delayed rectifier current is quantified in these two panels by measuring their mRNA copy number. And you can see across many different cells, so each bar here is a different cell, G, M, IC, LG, LP is the cell that was modeled in this paper. You can see that the expression level of mRNA can vary 3 to 5-fold depending on the cell type. And this suggests that within biological systems, there is a diversity that matches the diversity we observed in this modeling paper on the left. So the point is that this problem is something that's actually in biological systems. So even if you could find there is no quote-unquote perfect set of parameters, and that really makes the problem of modeling these complicated conductance-based models very difficult, and it's an unsolved problem that we need to continue to think about going forward.